Hello, I'm going to introduce our poster introducing the Breath Biopsy VOC Atlas, a list of microbiome-associated VOCs for breath-based biomarker discovery for this presentation for the 2024 ACG conference. Volatile organic compounds, or VOCs, found in exhaled breath can offer a promising route to safe, non-invasive diagnostics in multiple disease settings. Some of these VOCs are linked to the GI microbiome and could provide insight into disease development. The complex and unknown composition of the breath matrix makes identifying and validating on-breath VOC biomarkers a significant challenge. Therefore, we are developing the Breath Biopsy VOC Atlas to provide a comprehensive catalogue of VOCs identified in exhaled human breath. This atlas can be used to support the development of breath VOC biomarkers and diagnostics. For Project 1, 94 breath samples were collected from volunteers, and for Project 2, 76 breath samples were collected from healthy pre-diabetic and type 2 diabetic volunteers. All breath samples were collected using Alzo Medical's Breath Biopsy Omni-Sample Collection and Analysis platform. A paired equipment blank sample was generated by the sampling the room air immediately before each breath sample collection to compare with breath samples to distinguish VOCs originating from the background and those truly present in breath samples, which is also known as on-breath VOCs. 1,471 total features were identified in the breath samples, and 328 of these are classified as on-breath by 3 analytical metrics. Currently, more than 100 on-breath VOCs have been identified using comparisons to purified chemical standards and been added to the Breath Biopsy VOC Atlas. These compounds include aldehydes, alkenes, alkanes, alcohols, phenols, and many more. These categories will expand as additional VOCs are added to the atlas. Dietary fibers, which cannot be processed by humans, are fermented by microbes in the intestines, resulting in the production of VOCs such as short-chain fatty acids. These VOCs have roles in several signaling contexts, including the central nervous system, immunity, and inflammation. Other interesting microbiome-associated VOCs present in the Breath Biopsy VOC Atlas are related to inflammation, such as indole and isoprene. Indoles have been linked to maintaining the biological barrier of the human intestine. The results of this study highlight the power of the VOC Atlas and what it can bring to gastrointestinal research. The Atlas can support future breath studies, providing reference range of key on-breath VOCs and various cohorts, including disease and healthy populations, alongside insight and scientific context to the identified compounds to enable the confident selection of candidate biomarkers. Thank you very much for joining me today and hearing more about our poster. I hope you enjoy the rest of the conference.